Assalamualaikum. Today we are going to start with chapter number 39 and 40 of microbiology और इसमें हम important viruses करेंगे which are related to GIT tract. ठीक है. So first of all there's measles virus. Measles virus causes measles, panencephalitis and maculopapular rash. Features क्या होते हैं? It's enveloped virus, single stranded RNA and it has got RNA polymerase in it. Habitat and transmission is because of the respiratory droplets. Pathogenesis, it basically invades the upper respiratory tract from where it goes to lymph nodes and it involves skin, characterize a maculopapular rash on face. Lab diagnosis, it's because of the viral DNA. Treatment is vaccine given in combination with mumps and rubella. The vaccine is termed as MMR. Next, there is rubella virus. Rubella virus causes disease that is known as congenital rubella syndrome and it affects the neonates and they affect CNS and CVS. Features are it's an enveloped virus, it's single-stranded RNA and it has got no polymerase in it. What is habitat? Of course, the respiratory droplets and also it transfers across the placenta. Pathogenesis, it first goes into the nasopharynx where it goes to lymph nodes and then it involves skin and it also causes the congenital malformations in fetus. What are the lab diagnoses? Lab diagnoses are PCR assay because it's a virus. We can do PCR and also IgG antibodies. The reason these antibodies they help to diagnose rubella. And the treatment is same, of course, MMR vaccine. Then parvovirus B19, it causes slap cheek syndrome. This thing is very much important as it has frequently come in MCQs. Slap cheek syndrome, anaplastic anemia, arthritis and hydrops fetalis. These are the diseases caused by parvovirus B19. It's non-enveloped single-stranded DNA virus and habitat and transmission is same as of the rubella virus. What is the pathogenesis? The virus infects erythroblast and it causes rash on skin as well as arthritis and edema. Lab diagnosis is PCR assay. Next is norovirus. Norovirus causes gastroenteritis. Features are non-enveloped in single-stranded RNA. Its habitat is via fecal-oral route. Pathogenesis, infection limited to only mucosal cells. And then there's lab diagnosis, it's PCR assay and there are no neutrophils or red cells in stool is the diagnostic feature of the norovirus. What's treatment? No antiviral drug treatment. Basically we have to treat the symptoms. So we will treat diarrhea with fluid and electrolytes. Next is rotavirus. Rotavirus causes gastroenteritis. What are the features? It's snake double layered capsid. RNA polymerase is present. Habitat is same. Pathogenesis, it's limited to GIT tract, especially small intestine. Lab diagnosis, PCR treatment consists of two vaccines. One is rotavirus stereotype and other is live vaccine. Next is poliovirus, very important virus. It causes poliomyelitis and aseptic meningitis. Features are its naked genome RNA produces viral proteins. Habitat is same. Pathogenesis, it involves pharynx, then it transfers to lymph nodes and blood and also involves CNS and it causes muscular paralysis. Lab diagnosis is isolation from stool and detection by CPE and also it is characterized by raise and antibodies titer. So treatment is basically no treatment, there is prevention which is of polio vaccination. Cockaxi it causes meningitis, hepangioma, myocarditis, pericarditis and it also involves it causes hand foot mouth disease and juvenile diabetes. Features are it's naked and single stranded RNA. Pathogenesis, it involves oropharynx from where it transfers to blood and different organs. Lab diagnoses are PCR assay, CPE and raise in antibody titer. This is all about chapter number 39 plus 40 of microbiology. Thank you.